Welcome everyone to the third episode of the GIFT webinar series called Women Up to No Good. Women Up to No Good is a series where we ask the questions that matter most when it comes to female sexual and reproductive health and how research and innovation can improve this. This webinar series is an initiative of the GIFT project currently being led by the University of Cape Town. In the chat box, we'll post a link to the GIFT website in case you're interested to learn more about the GIFT device and our team and what we're working on. I am Professor Joanne Passmore, for those who joined a little bit late, in the Division of Medical Virology at the Institute of Infectious Disease and Immunology and Molecular Medicine at the University of Cape Town. I have the pleasure of being today's facilitator for this third episode of the GIFT webinar, Women Up to No Good. As we approach World Microbiome Day, which is the 27th of June next week, we will delve into socially responsive and innovative approaches to understanding and treating bacterial vaginosis, or BV. It's a condition affecting the delicate balance of the vagina's microbiome. This session promises to shed light on the importance of understanding what constitutes normal for each of us, what the optimal vaginal bacteria for health should be, explore the potential of complete or curated vaginal microbiome transplants as a sustainable solution to bacterial vaginosis, and discuss the promising future of vaginal probiotics. Our aim for this dialogue is learning, reflecting, and connecting around this common objective of moving women's reproductive health forward. For this webinar episode, we've invited three remarkable scientific leaders in the female sexual and reproductive health space, and who we believe can effectively lay the foundations for these important discussions later on. We will ask each of them from their different standpoints about their work in this space and their efforts at empowering women to take charge of their sexual and reproductive health. So without further ado, I'll just introduce you to the format of the webinar. It's got a relatively structured format. We'll give each of our three experts a chance at the beginning to reflect on today's topic, and then we will move into an open question and answer session where you're welcome to ask questions via the chat box. Also be aware that the session is being recorded and will be available on the GIFT YouTube channel. This is also where you can catch up on other discussions that we've had in this series that you may have missed. To ensure that you don't miss out on the upcoming conversations we have and other important announcements, please sign up for our e-newsletter and stay tuned for our GIFT media channels. We're excited to have such a good turnout today. We've had over 100 people register for today's webinar. Please continue to introduce yourselves and engage with each other in the chat box. And please don't hesitate to use the chat box for any questions at any time through the webinar. Questions for the panelists can be added in the chat box, and we'll address these to the panelists later in the session. So today, I'm pleased to introduce our panel of experts. Professor Sarah Lebeer is professor at the University of Antwerp, who has studied beneficial microbiome, focusing on lactobacilli and their role in health. Sarah spearheads the ISALA Sisterhood Project, an award-winning citizen science initiative now involving sister projects in many different countries in Europe, South America, and I'm very pleased to say Africa. These sisterhood projects aim to inspire and support worldwide research in women's health and the microbiome. And Sarah's presentation will focus on the importance of women owning their vaginal health and the power of citizen science. Our second panelist is Dr. Caroline Mitchell, amazing physician scientist director of the vulvovaginal disorders program, and associate professor of obstetrics, gynecology, and reproductive biology at Massachusetts General Hospital in Boston. With both clinical practice and research laboratory components, Caroline's research focuses on basic science and translational research. She leads the groundbreaking vaginal microbiome transplant study, which we'll hear a little bit about today, hopefully, called the MOTIF study, which is funded by the NIH, which explores the use of vaginal microbiome transplants to prevent recurrent BV. And last but very not least, Dr. Desebu Putlani has joined us as our third expert. She is site director and reproductive health clinician at Caprice's Vulindlela Clinical Research Site in rural KwaZulu-Natal. She is a South African principal investigator on a study that is testing an exciting new strategy at shifting the vaginal microbiome in women with bacterial vaginosis. And this is called the Vibrant study, or the live biotherapeutic randomized trial. Desebu will share insights from Vibrant, a clinical trial testing the efficacy of vaginally administered tablets containing Lactobacillus crispatus, who's everyone's favorite bacteria, for preventing bacterial vaginosis. So I would like to invite each of our panelists to start this discussion with a brief scene setter by giving some insights into this topic based on their background and experience. And up first is Professor Sarah Lebeer. And then we'll go to Caroline and then Desebu. But over to you, Sarah. Thank you.
Thank you very much for this very kind introduction and also this very nice invitation, because for me, the GIFT project is really like a very nice example for the whole world, how you can advance women's health with vaginal microbiome research. I will quickly give an introduction to our ISALA project. It's a Belgian project where we combine vaginal microbiome research with citizen science. I'm not a clinician, I'm a basic microbiologist, and I study the human vagina as a very important, for me, it's the most important microbial organ because it's important for women's health, our reproduction, but also our well-being, and also the well-being of our partners and our children. And what's so unique about the human vagina is that it's unique for humans. So it has a very unique microbial composition. And under healthy conditions, what we already know now is that it's mostly dominated by lactobacillus species, and these lactobacillus species are my favorite bacteria as a microbiologist. But because it's so unique for humans, it's very difficult to study. You cannot use animal models. It's very difficult to use cell line models and so on. So we actually need human beings for your study. Even if, like me, you're not a clinician and you will want to study the fundamental aspects of the microbiome. So that's why we adopted citizen science in our research. And I think many of you are aware of the story of Henrietta Lacks. And it's actually, for me, it's like an example how you should not collaborate with women for science. As so her cells were taken without her informed consent. They're used across the world by companies. They make billions of money. And it's done in a really not ethical way. So we want to ask women to donate samples and give us information, but only on a collaborative and co-creational way. So we don't want to just treat women as sample and data donors, but we want to interact. And I want to quickly show you today that even for this fundamental part, if you research and you involve women and ask women to also give ideas on how you have to run your research, that it's not just, that it's also high quality research and that it can also be published in good journals, even if you spend a lot of time on communication and so on, and it's also relevant for impact. So in Belgium, we named the project ISALA because Isala van Dis is the first female physician in Belgium, more than 100 years ago. She was actually not allowed to study medicine in Belgium. She had to go to Switzerland to study medicine. Then she came back to Belgium. She wanted to open a practice that was not allowed. They really had to change the law. And then finally, she was able to open her practice. And her first patients were actually a big female population, and reproductive health and sexual health were very important. So for us, it was a very important role model, and we named our project after her. As I already told, I'm a basic microbiologist. So to study the vaginal microbiome, we have some clear goals. We want to study, actually, we want to better understand what is health. And I think the whole field needs to have a better understanding what is actually the composition in healthy women, because the field is very much dominated by research on problems and infections, problems such as bacterial vaginosis. But if you want to better understand these conditions and want to develop better treatment options, you also need to better understand the reference and the comparison. And for that, so you need interaction with citizens. And I think many of us, all women, but also our partners, we all agree that we want to better understand what are the life course events that matters, what are lifestyle and environmental factors that do have an impact. And then as microbiologists, it has also already been mentioned, we also hope to help women across the world with innovative therapies based on microbiome research. So, and for that, if you want to develop, for example, a novel probiotic, you need to have access to beneficial bacteria from healthy women. So you also need to collaborate with women. So from the beginning in our project, we said if we want to have all these samples and material from women, we also have to give something back to the women that donate samples, but also to the society in general. And we realized soon that doing a project on the vaginal microbiome is actually a very good opportunity to also break taboos on vaginal health with input from citizens. So we spend a lot of time in communication with our citizens and ask them which taboos should we break. And these are some examples from Belgium. For example, there's now a lot has been done on menopause, menstrual hygiene products, typical other vaginal complaints. We did some projects on the Ramadan or fasting behavior. And on the other hand, we also want to use a project to empower women to take intimate samples, to take their health into their own hands, but also to stimulate co-creation and ask input from women with diverse backgrounds on which aspects we should focus on in our research. So this is a very scientific project, but with the whole team, we find it very important to have a nice communication strategy with a nice logo, with a nice sampling box. And the sampling box should actually almost look like a gift box. So we also spend time on making, for example, also these tote bags, because the participants to our project, they should really feel themselves honored, and we want to properly acknowledge them. We also have a very nice website, isale.be. It's in English and different versions or in different languages, and on Instagram and also Twitter or now X and so on. And thanks also to my team making everything possible. So I will not go into detail of the results, but when we were preparing the project, we did all the effort to reach 200 women in Belgium, and everybody was telling us that it was way too ambitious. We would never reach these high numbers. But when we launched the call, it was actually the opposite. So within 10 days, we had participants, and the B panel is a map of Belgium. It's the pink part is the Flemish-speaking part. It was allowed to participate. And as if you can see, almost every little village in Flanders did participate within this call of 10 days. So we had a lot of participants willing to participate, and our oldest participant was even 98 years old. So for us, it's an example that women's health matters to all women, and they all want to know more about the vaginal microbiome. This is an open science project, so it's a citizen science project. It has a disadvantage that it's not a population-based studies. So in a way, our diversity was not ideal, but we're still proud that we have some quite diverse population and even women living below poverty level. And we are now working with focus groups on how to even increase our diversity. We spend a lot of time in communication, and this is an example of an infographic we then made for our participants. For example, one of the first very intriguing results was that if we did the effort to sample the vaginal microbiome outside the clinic, so we asked women to take samples in their privacy, or in the privacy of their own bathroom, then we did observe a lot of lactobacilli or lactic acid bacteria. And it was much more than when we did the sampling campaign in the hospital even. So for us, it shows that if you do the effort, you can really reach to healthy women and have a good sample source of healthy bacteria. We find a lot of new associations with lifestyle and diet, so very interesting avenues for further research. We have a huge sample collection. And what's also very nice now is that actually bottom up by very motivated researchers across the world, we now also have international sisterhood projects in many different countries across the world, and we also have new avenues for new research. So I don't have time to go into detail, but I want to show you this slide because I'm very proud, and it was actually not planned in the beginning, but it's so nice to see that many women and men across the world are motivated to start their own sisterhood projects across the world. For example, in Africa, we are trying to collaborate with the people from the GIFT project, but also we have this project in Cameroon, and it's actually bottom-uply organized by Josiane. She was a PhD student in Cameroon, and she did a citizen science approach in her capital, but also in the rural areas. And she really reached out to women and the male head of the villages, and she was allowed, they did a sampling campaign with the motorcycles, and the first analysis are recently done. So we have now also a new ID of the vaginal microbiome in that region. And then another big sisterhood project is in Peru. I'm also excited about that because I will have the opportunity to visit them. They are sampling already in the highlands, in the Amazon region, and in the capital. And what we learn from all these projects, when the effort is really done to sample healthy women, that you get a very nice, intriguing, diverse map of the vaginal microbiome across the world. And maybe as you can see on the names of the different projects, what's also nice to see is that every country or every research group also choose their own name for the project after a strong role model in their geographical region. So with that, I would like to end my short presentation and introduction and give the floor to the other speakers. This is just a quick slide to acknowledge the whole team, which is very motivated and enthusiastic. So we are a team of microbiologists, gynecologists, GPs, but also communication specialists, legal experts, sociologists, and so on. And that's also very important if you want to run a project like this. Multidisciplinarity is key. So thank you very much for this invitation.
Thank you so much for that inspirational talk, Sarah. You do know I'm a very big fan of the SALA project and what you're achieving. So onwards and upwards, and continue the brilliant work. So with that, I would like to open to Caroline Mitchell and hand over to her for the presentation.
That is a hard act to follow, but I will try. Thank you so much for the opportunity. So I'm going to talk to you today about the study that we are running. And I'm going to start talking just briefly about bacterial vaginosis, which many people in the webinar probably already know about. But this is a condition characterized by the absence of those beneficial vaginal lactobacilli that everyone loves, and the presence of many different kinds of anaerobic bacteria. Clinically, this can cause discharge and odor that can be very distressing for individual women, and it is associated with a twofold, at least, increased risk of adverse reproductive outcomes like preterm birth, HIV acquisition, HPV acquisition, and persistence. And so this is why it is the focus of a lot of work across many disciplines, both for the individual patient level care, but also for the public health implications. Treatment for bacterial vaginosis is antibiotic-based and has not really changed since the 1980s. These are the STI treatment guidelines in the US recommendations, and although we've added more nitroimidazole antibiotics, there's still just 2 categories of antibiotics, and that treatment is associated with a 60% recurrence in the first 6 months after treatment, which, given the tools that we have these days in science, is really unacceptable. We should be doing better for women. So why is there this recurrence? When we do antibiotic treatment for bacterial vaginosis, you'll see we start with this sort of diverse community, and we do reduce the levels of those BV-associated bacteria with antibiotics. And what we're left with is a community of primarily lactobacillus inners. You've heard all of us say how much we love lactobacillus. Lactobacillus inners is not one of our better lactobacillus. I would say it's probably more neutral. It's not bad. It is a lactobacillus. It does make lactic acid, but it's not great. And what we would rather have would be a vaginal microbiome dominated by Lactobacillus crispatus, which is what we think of as the most optimal lactobacillus, no matter where you are in the world. So how can we promote this Lactobacillus crispatus? Because it does not usually colonize after antibiotic treatment. So one product that has been studied for the past decade is a single species, Lactobacillus crispatus, single strain, so just one type of Lactobacillus crispatus, vaginal live biotherapeutic or probiotic called Lactin-V. And that has been studied in clinical trials and shown to significantly reduce the recurrence of BV after antibiotic therapy compared to placebo. However, the recurrence rate at 6 months is still around 30 to 40%, which is still way too high. One alternative would be to still use Lactobacillus crispatus, but multiple types of Lactobacillus crispatus. And that's what Desebo is going to talk to you about with the Vibrant study. But the third option is to say, well, why don't people have Lactobacillus crispatus? And why doesn't this single strain product of Lactin-V work as well as we would like? Maybe it's because Lactobacillus crispatus needs friends, needs a community to support it, and we're just not putting in everything that we need. The question then is, well, okay, what are the friends? What else should we be putting in? That we do not know. So the idea behind vaginal microbiome transplant is just let's put the whole good community in, and that will help us figure out what is necessary. So this has been tried before in at least 2 studies, and a third that will be published soon, I think, separate from ours. The first case series was from Israel. It was 3 donors, 5 recipients. And of those 5 recipients, all of whom had highly recurrent bacterial vaginosis, that nothing had worked, not antibiotics. It just kept coming back over and over. 4 of the 5 people ended up with a Lactobacillus crispatus dominant vaginal microbiome, although 2 people it worked right away, 2 people required multiple doses of transplant, and then 1 person it really did not work at all. There was also a case report of 1 donor, 1 recipient, and there was a very clear shift in the microbiome from BV-like to Lactobacillus crispatus dominant, and that study demonstrated clearly that the Lactobacillus crispatus was in fact the Lactobacillus crispatus from the donor, suggesting that this really did work to transplant a microbe. So that brings us to our study motif, which is the first randomized trial of vaginal microbiome transplant for recurrent bacterial vaginosis. It is 1-to-1 randomization with placebo. This is my co-investigator, Doug Kwan, and we are funded by the NIH. So one of the most important things when you do a study like this is safety, because we are transplanting human material. And so our donors undergo really extensive screening, including questionnaires that are based on blood donor questionnaires and cover risks for things like Zika and monkeypox, because these are reproductive age women who could potentially get pregnant. So we want to know about that. Our donors agree to be sexually abstinent for the entire period of donation, and we test upfront for multiple sexually transmitted infections and other things like CMV, which a primary infection could be associated with neonatal congenital deafness if people are pregnant. So again, people are not allowed to be pregnant in the study, but one never knows. So we're trying to be as safe as possible. And 95% of donors screen out. So we're very strict. If people meet all the criteria, they then provide donations over a maximum of 45 days using this disposable menstrual cup. They'll put it in the night before, wear it overnight, and drop it off at the lab in the morning. We test each individual donation for high-risk human papillomavirus, the HPV 6 and 11, which cause genital warts. For folks with male partners, we test for semen and sperm to make sure that people are, in fact, sexually abstinent. And then we confirm in each donation that Lactobacillus crispatus is much more prevalent and abundant than Lactobacillus inners. And then when people finish, we test them again to make sure that we did not miss any acute infection. We test right at the end of donation, and we test for bacterial STIs at that time. And then a month later, we just do the viral serologies again to, again, be sure that we did not miss an acute infection. So that's a lot of work up front for the donations. And then our recipient protocol, we enroll only people with a history of recurrent bacterial vaginosis. They do not have to have BV when we enroll them. They just have to not have a normal microbiome with a Nugent score of 0 to 3. We provide antibiotic treatment with oral metronidazole and then 2 doses of transplant in the same week, but on non-consecutive days. And then we follow people for up to 6 months. And the way we deliver the transplant is very not fancy. It is just a transfer pipette and a speculum exam. And it's, again, either saline placebo or transplant material. So these pilot results, the FDA required us to do a safety pilot of 8 people. And these were presented at the Society for Reproductive Investigation meeting by Dr. Fatima Hussein in March of this year. The results I'm going to present. So each column here is an individual visit for an individual participant. Each color is a species of bacteria. The red triangle is when people got metronidazole. The gold arrows are when people received transplant. And Lactobacillus crispatus is in gold because it is the best. Lactobacillus iners is in green. Gardnerella and Adipobium, which are familiar to many people as BV-associated bacteria, are blue and black. And so the top row is our placebo participants. The bottom row is our transplant participants. And I think what you'll see is that there's this one person in the placebo arm that has gold, although it's never more than 50% of the community. Whereas at 1 month for our transplant participants, 3 of the 4 people have a majority of Lactobacillus crispatus. This last person did have this, but then she lost it by 1 month. And at 6 months, 2 of our 4 transplant participants have greater than 50% Lactobacillus crispatus. This third one, it's still present at a little bit, but it is no longer dominant. And 50% may not sound like a lot, but all of these people have highly recurrent BV, very similar to the Israel case series, where several of them said, I just don't go to the doctor anymore because nothing works. So I've given up and I'm just living with it. And so to us, 50% is incredibly, incredibly exciting. And so we are continuing on with a larger cohort, more donors. This was all material from a single donor. And I think the questions we have going forward are, do you have to match characteristics of the donor and the recipient? Is 2 doses enough? Does it look like we should be giving more? So there's tons of questions that we'll be able to answer going forward. And obviously this is impossible without a big team, and especially my co-investigator, Doug Kwan. And we hope that the future of BV treatment is bright. So thank you so much for your attention, and I look forward to questions afterwards.
Thanks so much, Caroline. I have enormous respect for the amounts of hard, detailed work you've put in before samples are even transplanted. So it's really impressive, the framework, and I know you've published that. So really just thank you for the team for putting all of that out into the world. And just a reminder for people to put questions for any of our speakers into the chat. So our last speaker for today, I invite Desebu Potlani from Caprisa to share her slides, and we really, really look forward to hearing about the important work that Caprisa is doing in this space.
Thank you so much, Joanne, for the kind introduction, and I'm actually very excited to be presenting on the Vibrant trial today. I'm presenting on behalf of the Vaginal Microbiome Research Consortium, and Caroline and I are actually working on this trial together. So this is a brief overview of my presentation today, and I'll get right into it. So we've learned that a lactobacillus-dominated vaginal microbiome, and more specifically a lactobacillus crispatus-dominated microbiome, is the hallmark of a healthy vagina and female genital tract. Caroline has already alluded to the fact that BV is characterized by an increase in diversity and quantity of species in the genital tract and the absence of lactobacillus colonization. There are high BV prevalence rates globally, and data shows us that African women are disproportionately affected as they're more likely to have diverse vaginal microbiota, and also they're more likely to have vaginal communities dominated by L. iners rather than crispatus when you compare them to European women. Genital inflammation that is associated with BV and STIs increases the risk of HIV infection in women. It increases the influx of HIV-susceptible target cells into the space, and it facilitates the acquisition of less infectious HIV variants, which is incredibly important in HIV hyperendemic areas like the one that I work in. Our site is located in a rural community called Vulindlela, and this is located in the KZN Midlands. It's got a population of approximately 140,000 people, and studies have shown that women in this community are 8 times more likely to acquire HIV, and it occurs about 5 to 7 years earlier than in their male counterparts. So in the graph at the bottom left side shows the HIV prevalence rates in the Vulindlela area and in the adjacent Edendale communities. And we see this high prevalence rates of HIV starting from around the age of 15 and peaking at around 35 to 39 years old. So my interest in the vaginal microbiome is largely tied to how it contributes to the to HIV acquisition in women. So as we working to find efficacious and effective HIV prevention technologies, it is important that we also focus on altering and optimizing the vaginal microbiome in women. So similar to what Caroline has presented, this is data that comes from one of our CAPRISA studies. And what I'm trying to show in this is firstly, the high recurrence rates that you find after treating patients with BV. Also, that in women that have BV, treating with antibiotics does not promote Lactobacillus crispatus dominance. So on these slides, women that were enrolled in the CAPRISA 083 study were treated with metronidazole after they being diagnosed with BV, and the vaginal communities were characterized at baseline, 6 weeks, and at 12-week at the 12-week time point. So not to cause confusion with the colors that Caroline had in her graph, but here we have Lactobacillus crispatus in green, because green means go, and in yellow we have Lactobacillus iners. From this, we see that at the 6-week and 12-week time points, there's a larger proportion of women that had vaginal communities dominated by iners rather than crispatus. So we need interventions that is able to promote crispatus dominance after treatment or even without treatment. What are some of the strategies to promote crispatus dominance? We've seen that commercial probiotics that are targeted at vaginal health don't contain the vaginal strains, the lactobacillus strains that are actually present in the genital tract. They also show minimal to no benefit after use. A possible effective strategy is to deliver exogenous L. crispatus intravaginally. As Caroline has mentioned, this strategy was studied in the Lactin-V trial, where they used a single strain of L. crispatus in a live biotherapeutic product. And although the Lactin-V arm had lower recurrence rates compared to placebo, recurrences were still high in this arm, and they increased with time. So this tells us that we still need interventions that are able to durably shift the microbiota to one that is dominated by crispatus. So how do we do this? We hypothesize that a product that contains multiple strains of L. crispatus may be able to achieve this. And how do we identify those strains? This slide shows 3 women that were identified from the CAPRISA 084 study, which was a PrEP demonstration study. These 3 women had stable Lactobacillus crispatus dominated vaginal communities at least 3 consecutive study visits. A similar exercise was done at UCT, at the first site in Durban and in the US, to identify some of these women and to identify their specific strains of L-Crispatus. From this exercise, we're able to identify and isolate 15 strains from women in the US and in South Africa. And these are the 15 strains that will be that have been used to formulate our product. From the 15 strains, we identified 6 that actually yielded maximal genetic coverage when you're doing metagenomic studies. This brings us to the Vibrant trial. So the Vibrant trial is a first-in-human trial looking at our 2 live-by therapeutic products, and it aims to assess the safety, tolerability, and colonization kinetics of LC106 and LC115. So this is our 6-strain consortium and our 15-strain consortium. The objective of our study is to assess safety, colonization kinetics, to assess the capacity of the L-Crispatus strains that are contained in the live-by therapeutic product to establish colonization, and to identify optimal dosing frequency and duration for future studies. The trial is being conducted at 2 clinical research sites, at the Massachusetts General Hospital in Boston, Massachusetts, and at the Caprisa Vulindlela site in KZN. Both sites will enroll around 50 to 60 women that have BV by Nugent and AMSEL criteria. Women that have been successfully screened into the Vibrant trial will be randomized to one of 5 arms. All study participants will receive a 7-day course of oral metronidazole as per the local standard of care. In the first arm, participants will receive 7 days of a vaginal placebo tablet that is similar to our active product. In the second arm, participants will receive 7 days of active 106. In the third arm, 3 days of active 106 and 4 days of placebo. In the fourth arm, they'll start using their vaginal product in the middle of the antibiotic course. And in the last arm, they'll receive 7 days of active 115 following antibiotics. Participants will then enter an off-product follow-up phase of the study, and at the 5-week time point, that's where we aim to do our primary analysis. They will then have extended follow-up and will exit the study after 12 weeks. In addition to the clinical trial, we are also conducting a qualitative ancillary study to understand the experiences of LBP users that are enrolled in the Vibrant trial. So our goal is to reduce adverse reproductive health outcomes and also reduce HIV risk in women. This first-in-human study is among the first steps along that road. Our long-term goal for the project is to find an intervention that can prevent incident BV in women, in people at risk, and hopefully we will be able to do that without antibiotics. We aim to create a durable and predictable shift of the vaginal community to one of L-Crispatus dominance, thereby improving reproductive health outcomes for all women. I'd like to thank our collaborators. These are all the institutions that are represented by the VMRC members. Thank you so much, Joanne.
Thanks, Disebo, and just to acknowledge as well the enormous amount of effort in getting that study approved by SAHPRA, and also just to give a shout out to the vision to have this going through 2 clinical trials in the US and in South Africa concurrently. I know we've motivated very strongly that South African women need this so dearly. So having that parallel process has been really amazing to watch unfold. So I would just like to thank all of our 3 speakers for sharing this data, also for the generosity of sharing work that hasn't been published. As each of you know, I am waiting for the outcome of the MOTIF study and the Vibrant study, and I watch with great interest as I see publications coming from the ASALA team. So just really thank you for that generosity. So I'm going to say we should get the discussion going, and we can put a few questions to the panelists, and I just wanted to thank them also for being mindful of time and giving us the opportunity for this Q&A time. And please just feel free to continue adding questions to the chat. Okay, so we are going to, as much as I wanted to hog the whole discussion, we are going to take questions from the audience first. And we have a question here from the audience. It's a question for Caroline, saying the findings you presented are amazing. I'm just curious, have you looked at the absolute abundance of bacterial species following this vaginal microbiome transplant compared to placebo, which I think is a very important question.
We have, so we've done total 16S qPCR, and we've done qPCR for Crispatus, inners, Gardnerella, and then I think Adipobium at this point, or Fannyhesia vaginae, which it is now. Those results are not published. The one teaser I can give you is that that one person in the placebo arm who did have some Lactobacillus crispatus, the total quantity by qPCR for that person was 3 logs lower than the median concentration for the folks in the transplant arms. And so although it was there, it was not there in high abundance for that person. So more to come with the publication that hopefully by the end of the summer will be coming out on the pilot results.
And thanks, we'll be waiting for it. That question was from Chantel Klaassen Weitz, who is a postdoctoral fellow actually here at the University of Cape Town. So thanks for asking that, Chantel. And then we've got a comment and a question from Professor Lenine Liebenberg, who is sitting at the beautiful Stellenbosch University. She says, "Hi, Sarah. What a wonderful social and scientific initiative. Besides the swabs used for understanding microbial composition, as you described, does the project also conduct research to better understand associated microbe metabolic functions or other indicators of the local immune environment?"
Oh, thank you very much. That's a really good question and relevant question. So yes, we have. When we started the trial, I thought, okay, we will do the home sampling, and in Belgium it was also during the summer, so we asked women to take the samples at home. We did a whole pilot study so that it could be transferred at room temperature. It was a very hot summer, so we thought, okay, it was possible in high temperature, and then we double checked that we could still culture Lactobacillus crispatus and inners from all the samples. Again, a pilot study that was also possible within, the samples could be transported around I think up to 2 weeks, and it was still fine. And then the third step, then we thought, okay, if we are going to ask women to take samples, and we will not ask it too many times, but maybe immediately they can also take a swab for metabolomics and cytokine analysis. And we did also the metabolomic analysis. The first thing we actually checked was indeed the impact of the transportation time, whether that would be the major signal. And we did that together with EMBL in Heidelberg in Germany. And actually it was not the biggest signal, so we could do the metabolomic analysis, and we have now the first analysis in that. It's really amazing to see the different patterns in metabolomics signals between, for example, Crispatus dominated one and a Prevotella in our case is totally different. And we, for example, we also run a whole panel. I don't know whether you have, the O-link panel, but you can really screen a lot of cytokines and immune markers simultaneously. And it also worked perfectly fine on our citizen science samples. So what we are now trying to do is take as much analysis from 1 or 2 single samples so that we don't need to bother women too often, but that we can really do the basic fundamental science in real samples, so not just having to run just cell models or mouse models, which are not so reflective of the real vaginal community.
Thanks for that. So before I take, there's a question in the chat, but I just wanted to, you touched on it, and this is a question for Desebu from me, but I know one of the strengths of the work you do in Vulindlela and your team is the individual interviews and the qualitative studies that really are essential with products that are delivered vaginally. And I wondered, from the experience within your research and pre-exposure prophylaxis studies you've done where you've looked at topical products, like, can you just give a little bit more detail on how you're approaching the qualitative stuff? Because if we make something women don't want to use, we might as well just stop.
I completely agree with that. We've learned a lot from the HIV space. And part of my passion was to do the qualitative work alongside the clinical trial. So not for us to get to the end and say we have a product that is amazing and that works, but a product that nobody wants to use. So some of the questions that we're including in our IDIs are speaking to about disclosure of use, disclosure of participation. We know that women still, hold in high regard their partners' opinions, specifically related to reproductive health. So disclosure is an important one, but also whether they've used a vaginal product before of any kind and what the experience with that was. In the Vibrant trial, we're also using an applicator to deliver the product. And we're also asking about their experience with using the applicator specifically. How would they have liked to, have inserted the product? The duration of product use, was that acceptable? If they'd be willing to use the product again, if they think their peers would be willing to use the product. So we're really trying to, gather information from different angles that will ultimately inform what our next product and what our next clinical trial looks like.
Thanks so much for that. And just, I think that's an essential piece. And I know we're going to learn a lot from those qualitative pieces. And then somebody from Antwerp, Lenke Botstressel. Sorry if I said it incorrectly. I did try. Welcome, Lenke. So the comment is, thanks to the speakers for sharing their projects, all really important and exciting. And can't wait to hear more about the results. So this is a question for Caroline. In the group that received the vaginal microbiome transplants, can you remind us what they were asked, if they were asked to abstain for the duration of follow-up? And were there any other sexual behavioral practices that might have influenced the microbiome returning to non-optimal one or LN is dominant?
Yes, thank you, Lenka, for the question. So with 4 people receiving transplant, we can't draw any definitive conclusions for sure. I will say the one person in the transplant group who reverted to inverse dominance, she was the one person who totally abstained during the whole thing. Everyone else had some variety of sexual activity. So we're not asking people to abstain because this is real life. And in our larger study, hopefully we'll be able to assess how much that impacts. And we ask questions about, are you using any vaginal products? Are you bleeding? We're asking all of those questions to try and get at what might contribute to success or failure.
Amazing. Thanks, Caroline. And then as soon as it's going to have to be our last question, unfortunately, there's oh, sorry, it's Lenka from Australia, just to clarify. I assumed, sorry. Okay, so apologies for that. The question here is from Josiane Kenfak, who I know is very much a microbiome enthusiast. And if I'm not mistaken, I hope I'm not going to guess this wrong, but this is the amazing Josiane from Cameroon. The question is for Disebo, and she wanted to ask, well, she says that certain behaviors can increase risk of bacterial vaginosis, like being ill, hygienic habits, etc. So she would like to know if you're asking your participants to avoid these behaviors during the study.
Yes, so a very important question. Thank you for the question. So during the screening process, we do ask women about vaginally inserted practices, and if they're using any vaginally inserted products. And in the screening process, we do exclude women that have used a vaginally inserted product, or practicing any kind of vaginal cleansing, specifically like douching, before they enroll. So for 30 days before they enroll. And during their participation in the trial, we do ask them to refrain from using any vaginally inserted products, to refrain from douching. We do counsel them throughout their study participation, but we do also collect information about it. So if it has happened that someone has used a product, we'd rather know about it than not. So we do include it in our questionnaires, but there is ongoing counseling to refrain from using any other vaginally inserted product. Sexual activity, another important one. So we are also not asking participants to refrain or to abstain, but we are providing condoms, trying to encourage them to use condoms as much as possible, because as we know, condomless sex is more closely associated with BV and BV recurrences. So we counsel them in that respect. We provide them with condoms at the clinic as part of their little care package. And yeah, but we still collect information about it in our questionnaires about whether condoms were used and whether there's been any sexual activity.
So thank you very much for that. And I unfortunately didn't get to put any of my questions in, but I will hunt you all down separately. So I just wanted to thank the audience for putting the questions into the chat, for joining us today. I know it's a lot of really good colleagues from all over South Africa and other places too in the audience. So finally, I just wanted to actually express my sincere gratitude to some very busy people, Caroline Mitchell, Sarah Labia, and Desibu Putlani, for making the time to talk to us today, for sharing so generously, and for the significant contribution each of you are making to women's health. So I just wish you all the success in all of these endeavors. And the GIFT team would be very grateful to the audience if they would complete this link to the evaluation form for today's webinar. We're trying to cover topics that really resonate with the audience. So please share your thoughts and give us some feedback. And our next Women Up to No Good webinar is scheduled for September. So please watch the space. Keep an eye out on social media for more information about this. And lastly, there's our e-newsletter, which we're incredibly proud of, that tells you about all the developments that we're excited about and about future events, and that's in the chat box as well. So I would like to just end off with saying, for those who have missed any parts of the webinar or who know colleagues who weren't able to attend, a recording will be uploaded and into the GIFT YouTube channel, and a summary will also be available on the website. I just wanted to thank everyone deeply for your participation, and we look forward to welcoming you in the audience to our next webinars. And last but not least, just again, a really big thank you to the speakers for today for sharing this important topic with us. So goodbye for now, and have a good day further.